Great. Well, thank you so much. It's really quite an honor to be included in this lineup. So really happy to tell you about some of the uncommon solutions, I guess, that we're working on. And what I'm going to talk about is where I see the future of microbiome-based therapeutics going. And of course, the microbiome is the collection of microorganisms living in and on the human body. Right now, we don't actually have any microbiome engineered therapeutics out there on the market. What we do have is fecal microbiota transplantation, so fecal transplants. And these are being used fairly widely now for recurrent Clostridium difficile infection. So I'm going to tell you a little bit about that, but I'm going to cast it in the light of where we want to go, and I'm going to show you how we're using this data to help us design the next generation of microbiome-based therapeutics, which are going to be microbes that are isolated from human beings, but grown in pure culture and really look a lot more like what you folks and FDA would consider a drug. So maybe I'll start with a story of how I got into microbiome. So when I started here at MIT, I was really working more in the field of environmental microbiology, but it was kind of at the time of the very beginning of the human microbiome project at NIH. I was determined not to get into research on biomedicine because I thought there were enough people already doing that kind of research and not enough people studying microbes in the environment and things like that. But I got hooked up with this guy, Athos Bousvaros, a really fantastic doctor who ran the Pediatric Inflammatory Bowel Disease Center over at Children's Hospital. And Athos said, we've got a really interesting statistics problem here in trying to develop a microbiome-based diagnostic. And I said, statistics, that sounds interesting. And I'll let you kind of handle the clinical side. And so, Athos said, Eric, we have this problem. At the time that we started this study, there was about a 3-year lag time between the time that children, developed disease, inflammatory bowel disease, and the time at which they got an accurate diagnosis. Now, if inflammatory bowel disease develops later in life, it's a fairly simple diagnosis, but in a lot of young children, what will happen is they'll present with symptoms like depression and weight loss, and their grades are falling in school. And at that time, 3 years to get the accurate diagnosis because they get off, sent off to a psychologist or something like that to help with some of these other symptoms before they eventually make their way to a GI doc. And part of the problem is that, every 8-year-old kid that comes in with those symptoms, you obviously don't want to send out for a colonoscopy. So what would be useful in the clinic is a very sensitive but non-invasive diagnostic. And although there's some non-invasive diagnostics on the market, they tend to be more specific but not quite as sensitive. And so Athos had the idea of, let's take advantage of this new microbiome science. Let's sequence the DNA from the bacteria that are in fecal matter, and then see if we can predict disease that way. So he rounded up about 105 patients in the first pass, and I'm showing you sort of what those data look like. So all of these columns here are different patients, and so these are the control kids with Crohn's disease and kids with ulcerative colitis. And along the rows are the different bacteria, and so the darker colors means there's more of that bacteria in that particular patient. Okay. And the controls here are a really good control set because these were the kids who got referred to the clinic. So they had the type of symptoms that Athos would see on a day-to-day basis, but they ended up after their clinical evaluation, being diagnosed with essentially IBS, but not an inflammatory condition. Okay, so this is really, these aren't healthy kids. These are exactly the kids who we want to differentiate from the kids that actually have inflammatory disease. So if you look at this, there's no one bacteria here where if you saw that bacteria in a child, you would say this child has inflammatory bowel disease. The closest that we would come is maybe E. coli. Okay, and so we can see all of the kids that really have high levels of E. coli do have one of the two forms of inflammatory bowel disease, but that's not a very sensitive test, because if we said, we're only going to test positive the kids that have E. coli, well, we would only get about 10 or 15% of all the kids. We wouldn't make any mistakes, any false positives, but again, that's not what we're worried about, because all of these kids who test positive here are going out for a colonoscopy anyway. So we want a really sensitive test. We want to make a few false positives, but we don't want any false negatives. So we want something that we can adjust. And what we did is we took all the sequencing data, we combined it together using some machine learning algorithms, and we were able to take all of these weak predictors. Most of them look like this. So if we look at subdilogranulum here, you can see it's generally present in the healthy kids, much less abundant in the sick kids. Again, not a good predictor, but when we combine them together, we actually got a non-invasive diagnostic that was better than any of the commercial ones that were on the market. And what's really neat about this is when we look, as we continue to add more kids' samples to the diagnostic, it learns and it gets better and better. Okay, and so that's what's really got me kind of hooked on this field. So the very next year, I decided to take science into my own hands, and myself and my student collected our microbiomes every day. We collected all of our stool samples, put them in the freezer, and then later we did DNA sequencing to see how they evolved in time. And at the same time, we recorded every aspect of our lives. We recorded all of our diet, all of our sickness, travel history, things like that, 300 different lifestyle and behavioral factors. And this is what we saw. So this is my microbiome in 2009, and this is my student Lawrence David's microbiome in 2009. And the one thing that you can take away from this is there's variation day to day, but I could pick any day from my time series. You would know it uniquely as mine. Okay, so it's very easy to kind of identify it. So there's an enormous stability of long-term stability of the microbiome, and an enormous difference between any two people. I want to dive a little deeper now. So here's the same plot. So this is my microbiome. And now you get to see more of the bacteria. The bacteria are the rows here. And again, this is time. But I've normalized it so the warmer colors means there's more of that bacteria on that day. The cooler colors indicate there's less of that bacteria on that day. When I show it in this view, you can actually see there's a lot more going on than you even noticed on the last slide. So about 3/4 of the way into the study, I went out to brunch with a colleague here. I ordered some undercooked French toast, and then I got this salmonella infection. And of course, I went to the urgent care, and they said, "Oh, it's a virus. Just rehydrate." So I didn't get any antibiotics. Okay, now I have DNA evidence that was a misdiagnosis. But what happened was these bacteria here all disappeared from my gut. Some of them came back. Okay, but the majority were gone permanently. However, they were replaced with these bacteria here, many of which had never been seen in my gut until that salmonella event. And so this really got me thinking that, this is interesting because not only can we use the microbiome to make diagnoses, but we can think about re-engineering the microbiome. And if the microbiome is associated with health, what this tells me is that we can figure out what a healthy microbiome looks like and then go into patients that have issues, some dysbiosis in their microbiome, and we can figure out ways to re-engineer it. Okay, and so the paradigm is really simple. We get as many people as we can find. We sequence their microbes. We identify the bacteria by sequencing a particular molecular barcode, the 16S ribosomal RNA. We compare the community of organisms in healthy individuals and sick individuals, and that gives us hypotheses. And it gives us hypotheses that we can actually test in a clinical trial. If we observe that all of our sick individuals don't have a particular group of bacteria, Well, at the same time, when we're collecting samples from these patients, we're culturing those bacteria in a pure form, and we can go and we can grab them out of the freezer, we can lyophilize them, put them in a pill, okay, and then feed them to patients. And you know, I've gone from DNA sequencing to something that I can actually test in a clinical trial very, very quickly, okay? Sort of much quicker than I could if I were looking for a drug target and then trying to, you know, identify a small molecule and looking at toxicity. Toxicity, I imagine, is going to be fairly low because these organisms are probably natural parts of the human community, the ones that we're going to be putting back into patients. So that's the basic paradigm, and in terms of how would one make a drug that really works, I think the principles are a little bit different than the principles for making a small molecule drug. And in particular, I think there's sort of 3 different boxes that we need to check. One is engraftment. So most of the bacteria that we put into that pill are probably not going to colonize the patient, and we need to know the rules that determine whether or not a particular bacteria is going to wake up and actively colonize the patient. And we think back to Bob's talk about well, you know, how do we get drugs that continue to dose over time? We've solved that problem with the microbiome, because these bugs as drugs are going to grow, and they're going to colonize that patient, and they're going to persist there until we have some other intervention and remove them. But they're probably that's not going to work most of the time. We need to understand how can we choose bugs or engineer bugs so that they will colonize a particular patient. So we need to think about engraftment. Here, you know, all of these bugs were kind of kicked out of my ecosystem, which was otherwise very stable, and they were replaced here. So we know we can do it. There are certain things that we can do. Probably don't want to give all our patients salmonella infections, but maybe there's another way to engineer the system to allow for engraftment. The other is mechanism. This is the same with small molecule drugs. We can probably use really similar assays to figure out if the organisms that we're putting in have the desired effect. This is a mechanism from a Nature paper that suggests you know, maybe we could treat inflammatory bowel disease by upping butyrate production. And so you know, maybe our assay is something we can do in vitro, and we can see which organisms produce the maximum amount of butyrate from the same input of, let's say, dietary fiber or something like that. The last is kind of analogous to dosing, but I would argue that when we're talking about live therapeutics, it doesn't matter how much of the drug I put in the pill. That has almost no bearing on the concentration of that bacteria in the patient's gut. Okay, that's going to be set by a homeostatic you know, host microbe interaction that really is completely independent. As long as we put enough of the bacteria in the pill, the overall abundance is going to be set through these biological interactions. And so we need to understand that, because when we look at the abundance of microbes, some of them are very abundant, but it tends to have this power law distribution and this very rapid fall off, so that most of the microbes are present at less than 0.0001%. And so if our mechanism is to make lots of butyrate, and we get a bug that engrafts in 100% of our patients, and it makes tons of butyrate in vitro, well, if it's only responsible for 0.0001% of the biomass, it can't possibly substantially affect the amount of butyrate being produced in that patient's gut. So these, I think, are the general principles that are going to guide how we design microbiome-based therapeutics. Here's a particular case study, and this is really the only disease that we currently treat with fecal transplants. So a patient will come into the clinic, they'll get antibiotics for an unrelated condition, we'll clear out the gut microbiome, and if they're exposed to C. difficile, they may end up with a C. diff infection, especially after they've gotten antibiotics, and there's sort of this clean slate for the C. diff to grow in. So what do? We treat them with antibiotics again because they have an infection, brings them back to this state, And now about 30% of those patients will recur and they'll get a C. diff infection again. So of course we give them more antibiotics and we repeat the cycle. And after we've repeated the cycle about 3 times, they become classified as a recurrent C. diff patient. And it starts to become less and less likely that those patients are really ever going to respond to antibiotic therapy to clear that C. diff. Fortunately, we do have a cure. And so if we can inoculate these patients after antibiotics with gut bacteria from healthy individuals, there's about a 90% cure rate. So actually, it's a very, very effective drug. So a graduate student in my lab had a family friend who was infected with C. diff and had heard about the science, couldn't get it, ended up starting a nonprofit stool bank, which started out just extracting healthy bacteria from healthy individuals and mailing it to doctors around the country who were interested in this. And so that's Mark Smith. He started Open Biome sort of out of one of our biosafety hoods in the lab. It's now a 45 full-time employee operation operating out of Medford. And really has a clinical mission, but has contributed quite a bit to our research mission as well within the lab. These were our sort of milestones at Open Biome. It started in October of 2013. We hoped to treat 1 patient. We ended up treating 6. We wanted to treat 1,000 in 2014. We almost doubled that. In 2015, aimed to hit 10% of recurrent C. difficile infection. We hit that number in June. Currently, we're treating about 40% of all recurrent C. diff in the United States, working with about 600 hospitals, and we treated about 12,000 patients so far. But really, where we're really looking to grow is finding the next disease that might be amenable to a microbiome-based intervention. And so we think with the clinical network that we've built up through Open Biome, this is going to be, we're really well positioned to do that. So let me tell you a little bit, very briefly, about a study that we did with Libby Holman, Ilan Youngster, Jenny Sauk over at MGH, and then Ramnik Xavier and Dirk Jeevers at the Broad. So we had about 4 healthy donors going into 20 C. diff patients, about 90% recovery levels, and we got the DNA because we wanted to see what does a fecal transplant look like. And so on the top is basically the darker colors indicate there's more of that bacteria. So the columns here are the different bacteria. I'm not showing their names. But you can see this is what our patient looks like before the transplant. This is what our donor looks like. And this is what our patient looks like after the transplant. And so this is sort of the graphical view of a fecal transplant. It's really hard to predict whether or not a particular species is going to engraft or not. But we sort of have to do that if we're going to start to engineer the microbiome and engineer the next generation of therapeutics. So Chris Smiley, a very talented computer science graduate student, took this challenge on to take a lot of DNA sequence data from patients before transplant and from their donors and try to predict which species would engraft and which ones would not. He was able to use all of these input data to build a very accurate predictor of whether a species would engraft, and also if they did engraft, predict their abundance. And so this blue line indicates their predicted abundance, and we can see if we only choose bacteria above this cutoff, these are actually mostly quite abundant bacteria. So we could use this to design a drug by predicting the abundance of the bacteria after the transplant, so we don't end up with any bacteria that are only there at a fraction of a percent. This is sort of qualitatively what his model looks like. So you can see again that there's a really nice correspondence between what the patient looks like post-transplant and what the model predicts for that patient. That goes beyond, I think, what we can sort of do with the human eye. But of the factors that we looked at, which were the factors that kind of determined engraftment? So we looked at quite a few, and what it really boiled down to was knowing the genetic background of the bacteria, and then knowing the abundance of the bacteria in the donor and the patient beforehand. And a lot of the other things that we thought might make a difference, like oxygen sensitivity and things like that, had surprisingly little importance in the computational model. So how can we use this? Well, we start out with this whole community of bacteria here. And this is sort of everything that goes into a fecal transplant. And what we want to know is which are the bugs that we could grow up in pure culture and put into our pill to make something that looks a lot more like a drug to treat this disease. Well, we can first look at engraftment and only choose the bugs that are actually engrafting during a fecal transplant, and then we can look at abundance and only look at those bugs that are reasonably abundant and might contribute to some sort of metabolic mechanism. And so that gives us, within each of these clades, a smaller number of bacteria that from a practical point of view could possibly be the active ingredient in a fecal transplant. And then based on the disease that we're trying to treat, in this case, we're probably looking for inhibitors of Clostridium difficile, so closely related bugs. It really helps us narrow in on which of these hundreds or thousands of species might actually be the right active ingredient to start to make a therapeutic with. Because it's going to be very difficult to try to manufacture hundreds or thousands of strains. Timing, are we? Getting close. All right. So maybe I'll just go over really quickly. We got some additional information on these bugs, and we wanted to ask, are we sure that the bugs from our donor are really making it into the patient? So Chris developed a computational algorithm that can take all these fragmented DNA samples, and the output is really the genotype of the bacteria, so the precise genome sequence, and in how many different strains of each bacteria are in the patient. So here we can see subject 1 has 25% of this genotype and 75% of that one. And so that's the output from his program Strain Finder. And when we applied it to these data, what we found is here's one donor, and here's a species Ruminococcus. Our donor had 2 strains, and our patient at 3 different time points after the transplant picked up both strains of Ruminococcus, whereas the patient had none beforehand. Here's a patient with 4 strains, or a donor with 4 strains going into a patient. The patient picks up all 4 strains. Here's another species, F. prausnitzii. The donor had 4 strains. The patient had 4 strains. But then this new strain came up that wasn't in the donor material and wasn't in the patient. And so what we found is that once a species engrafts in a patient, that patient can start to pick up new strains of that species from the environment. In some cases, we saw that when the patient already had the species, they kept the species post-transplant. And sometimes when the patient already had the species, the donor species took over and wiped out the indigenous species. But overall, we found that either none of the strains will engraft or all of the strains will engraft. So it had this interesting all or nothing behavior. And so, in fact, more than all or nothing, here black shows patient strains, white shows donor strains, and red are these new strains that were neither in the patient nor the donor but came up post-transplant. And so what all this suggests to me is that really the environment itself is contributing a lot to these cases, and the environment of the patient's gut is deciding sort of what engrafts and what doesn't. And so I'll sort of leave you with this. I'll go back to these data. These are my own personal microbiome data, and a really terrific graduate student, Martin Poltz's lab, Dan Van Insberg, came to me and he said, Eric, I'm really interested in time series data, very interested in Clostridium difficile. I want to look for Clostridium difficile in your time series. I said, Dave, I'm a young guy, I'm pretty healthy. I definitely don't have C. diff. So he got my data, and it turns out I was dead wrong. So I didn't have any C. diff until I got this salmonella-induced diarrhea, and it created that environment that C. diff really thrives in. Once the environment was there, I picked up C. diff, and I've been asymptomatic carrier ever since. So last year, I took my 10-year-old son and my 7-year-old mother to the ruins at Petra in Jordan. We stayed in a Bedouin camp. we all got a terrible case of Giardia. So I went back to the urgent care again and said, oh, I feel terrible. He's like, yeah, you definitely have Giardia. Have you ever had C. diff? I said, that's excellent, because we're going to treat you with metronidazole, and that would be counterindicated. I said, well, that's very good antibiotic stewardship. I'm certainly not the demographic that I think would have had C. diff, but in fact, doctor, I sequence my DNA every day, and I can tell you that, in fact, I'm an asymptomatic carrier of C. diff. So we're able to sort of, again, use these data to this time make an accurate diagnosis at the urgent care. So with that, happy to take a question or two, and thanks a lot for your attention.
All right, questions? Everyone is ready for Nubar. Are you still looking for people to contribute to your data sets? Because this is an opportunity.
We're always looking, yeah.
So, the question is, how generalizable are these microbiome principles to other sites in the body? So you talked a lot about the gut microbiome, but you have obviously skin microbiome, ocular microbiome, nasal microbiome, all these other sites that have been implicated in health and disease.
Yeah.
And so being able to engraft in those sites and in those three boxes, like if we figure it out for the gut, would it apply to other sites? Or you think it's going to be very specific?
Absolutely, absolutely. I think the skin is a big one, and we know very, very little about how to engineer the skin microbiome. I think the vaginal microbiome is probably a great opportunity for engineering. The oral microbiome, especially around teeth and periodontal disease, is probably another really great opportunity. Most of these we know almost nothing about in terms of engineering. There's a lot that's been done in terms of surveying what bacteria are there and which ones might be good, which ones might be bad. Very little about how to engineer them.
I wanted to ask you if the research on the microbiome has in any way tempered the use of broad-spectrum antibiotics. I mean, are there now more risks that we didn't understand before when doctors give a lot of antibiotics to patients?
Certainly there's a lot of epidemiological data connecting early life antibiotic use to conditions later in life. I think probably a lot of that has been spurred by interest in the microbiome, but I don't know that there's anything directly in the microbiome to, that's more mechanistic that would explain, what the connection is. But I think certainly the increased focus on microbiome and its role in shaping immunity has certainly helped in raising awareness.
I don't know if you mentioned it, but you're saying the all or nothing for engraftment. Do you know anything about, is it differences in the host that may be driving this, or is it the microbiome pre-treatment? Do you have any indication why you have all or nothing phenotype?
Yeah, so it could be. It could be genetics, could be immunity, could be diet. I suspect diet certainly plays a role. I'd be very surprised if diet wasn't part of it. Probably all three contribute. I bet my guess would be genetics contributes less than immunity, just because we see not only an all or nothing behavior, but an all or even more than all or nothing. And to me, that sounds more like the disposition of the immune system toward bugs with those particular antigens is changing, right? Yeah, we don't really understand it.
Fancy making any predictions about the microbiome's role in allergies, cancer, other immune diseases?
Well, there's some fun papers out on, I shouldn't say fun, some exciting papers out on the role of microbiome as sort of an adjuvant to checkpoint inhibitors and things like that. Some really interesting mouse data. So Yeah, there's a lot of areas where I think microbiome-based interventions are going to start emerging. And there's already companies kind of working on all of them. So.
Okay. There's two doing similar things that you're doing, right? But they are for-profit companies.
Oh, there's more than two.
More than two?
Yeah, there's at last count at least 60. There's 60 companies out there making microbiome-based therapeutics.
That's
so.
amazing.
Yeah.
Did you hear? You must have heard the Signal podcast that interviewed your postdoc, and they were talking about some of the companies. No?
Oh, I didn't.
You should listen to it. It was a great podcast by Meg Tyrrell and Luke Timmerman.
Oh, super. Yeah, I'll have to check it out.
They can pay me later for my plug. Do you have one more question?
Did you get ethics to study yourself?
Oh, yeah, of course. Yeah. No, we're all strictly legit here. Yeah.
It's MIT. All right, so let's thank Eric and Bob and Terry one more time.